Welcome viewers. Welcome to the third lecture on classification of viruses. In our first two lectures on classification of viruses, we have seen that initially the viruses were classified on the basis of the diseases they caused. This classification was a problematic one as there are many viruses that do not cause any disease at all. The second method of classification of viruses was based on their host range. On that basis, they were classified as viruses that infect bacteria, plants and animals. In this case, we have seen that there are certain viruses that can infect both plants and insects. So, just by studying a virus that is infecting a particular host, we cannot have an idea about any other virus that is infecting the same host. So, the third methodology for classification that was opted was classification on the basis of the morphology. On the basis of the morphology, we have seen that they are classified as having helical symmetry or having cubical symmetry or having icosahedral symmetry, or they were called as having complex symmetry bearing viruses. But this classification has nothing to do with the molecular biology of the virus, or their mode of establishment inside the host is not being mentioned in this classification. So, the fourth classification that was used was called LHT classification, which was discovered by Loff. Therein we have seen that a proper taxonomic hierarchical classification system was opted for classification of viruses. This classification system was facing certain problems, and to overrule it, there came Baltimore classification system, which was utilizing the nucleic acid content, or the method of replication of the virus was given the priority rule for classification of the viruses. Baltimore classification system classifies viruses into 7 different groups. This classification system was quite okay, but still we had certain problems. Therein we have seen that the group 1 of Baltimore classification scheme of viruses comprises of T2 bacteriophage as well as variola virus. These two viruses are diametrically opposite, but still they are clubbed together into the same group as their replication mechanisms were similar. So, the Virologist Society was facing a serious problem, and it was looking for a classification system which will overrule all these drawbacks, and taking help of such a classification, any virologist who is searching for a new virus or who is working on a new virus can place his newly designated virus into a proper place. Then in the year 1966, International Committee on Taxonomy of Viruses, called ICTV, was formed. It is a committee of Virology division of the International Union of Microbiological Societies. The speciality about this classification system or this unit was that they used to meet after every 4 years, and they reanalyze the data that they have procured, and if necessary the places of the viruses were changed. They also developed a database called ICTV DB. DB stands for database. In this database, all the informations were uploaded in very different languages, so that virologists worldwide can get access into that database and can have a knowledge about the classification of viruses. Now, let's have a look at on what parameters the classification of viruses were done in ICTV. Number 1, the criteria that they used for the classification was the chemical characteristics of the virus. The second feature that was used was the nucleic acid content of the virus. As we all know that viruses can have either DNA or RNA in their genomes. So, this second point was taken into account. The third point that was taken into account was the replication mechanism of the viruses. The fourth point that was taken into account was the diseases that the viruses can cause. The fifth point that was taken into account was the vectors that are being utilized by the viruses for the mode of their communication. And finally, they have also acknowledged, or they have also taken into account the geographical conditions wherefrom they have isolated the virus. So it was a very scientific approach wherein all these 6 criteria was taken into account for the classification of viruses. Objectives of ICTV classification was to design an universally acceptable taxonomy of viruses. Next, they wanted to develop a taxonomic approach not only for the viruses but also for the subviral agents like satellites, prions and retrotransposons. They also were interested that whatever informations that are being put into the ICTV should be internationally acceptable to all the virologists who are working worldwide. To perform this, they created the database, as I mentioned earlier, that ICTV-DB was regularly maintained and it was upgraded time to time to keep the virologists working worldwide updated with whatever changes are occurring in this classification system. Now we will be discussing about the scope of the ICTV. ICTV system was classifying the viruses till the species level. It doesn't want to classify the serotypes, genotypes, strains, variants of the virus species. It doesn't take care of any artificially synthesized or virus that were developed in the laboratory was not assigned in ICTV. Next, we will discuss the one important point of difference between ICTV approach and International Code of Nomenclature for algae and fungi. Herein, for the first time, we will see that the orders and families were annotated with capital letters and italics, unlike the other systems. Herein, we couldn't classify the viruses at the level of kingdom, phylum or class. The first system of classification was the order. Next, look into the rules which were opted to classify the viruses in the ICTV approach. It shall be universally acceptable. Number 2, the hierarchical order that was maintained was order, family, subfamily, genus and finally species. Beyond that, they were not interested to look into. Next, then, viruses are put into species, the species are placed into genus, then the genera were placed into subfamilies or families, and finally the families were clubbed together into the orders. So herein we can see that if a species is not getting entry into a proper genus, but it has got entry into a family, then it is called as an unassigned virus existing in a family. There are cases wherein we have seen that certain genus could not be placed into families. So they are called as floating creatures. Next, we have seen that some families are classified together into orders, but for many, the family was supposed to be the highest class of classification. Now, if we look into the ICTV class of classification, we will see that it starts with order, which is indicated with the terminology ending with virales. Next, we have got families wherein they are written as viridae. Then we have got subfamilies which is mentioned as virinae. Then we have got genus which is mentioned as virus. Next, look into the report which was published in 2011, which states that ICTV comprising of 6 orders, 94 families, 22 subfamilies, it has got in its hold 395 genera and 2,480 species of viruses were reported till 2011. Next, we will talk about the orders of ICTV approach. It comprises of 6 orders. These 6 orders have been established in 2011. It comprises of Cordovirales, Herpesvirales, Mononigavirales, Nidovirales, Picornavirales and Thymovirales. Next, we will be discussing the basic features of these different orders and the number of families which are incorporated in each order. So, we have got 6 orders. The first order is called Cordovirales. It comprises of 3 families. Second order is called Herpesvirales. It comprises of again 3 families. Then we have got Mononigavirales. It comprises of 4 families. Next comes Nidovirales, which comprises of 3 families. Then we have got Picornavirales. It comprises of 5 families, and Thymovirales. It comprises of 4 families, and still there remain 72 families which couldn't be placed into any of the orders. They are still in search of new orders. Next, we will talk about the special features of these orders and name of the families that are present in these orders. So, let's start with Cordovirales. Cordovirales comprises of double-stranded DNA bacteriophages. It comprises of 3 families, namely Myoviridae, Cephuviridae and Podoviridae. Next, we will be discussing about Herpesvirales. Herpesvirales also comprises of double-stranded DNA viruses, but they infect eukaryotic hosts. That's why it is different from Cordovirales. It again comprises of 3 families, namely Herpesviridae, Alloherpesviridae and Megaloherpesviridae. Next, we come about Mononigavirales. Mononigavirales comprises of a non-segmented RNA viruses, which is of negative sense. It comprises of 4 different families, namely Bornaviridae, Rhabdoviridae, Filoviridae and Paramyxoviridae. Next, we will be discussing about Nidovirale order. Nidovirales comprises of 3 different families called Coronaviridae, Arteriviridae and Roniviridae. The special feature is it is a positive sense single-stranded RNA genomes. Next, we will be discussing about Picornavirale order. Picornavirale order is comprising of a small single-stranded RNA that infects a varieties of plants, insects and animal hosts. So, here we can see different families are clubbed together which can infect a wide range of hosts. So, here we have got 5 families, namely Calciviridae, Dicystroviridae, Picornaviridae, Sequiviridae and Potiviridae. Next, we are coming to Thymovirales order. This Thymovirales comprises of plus sense or single-stranded RNA viruses that infect mostly plants. Herein, we have got 4 different families called Alpha Flexiviridae, Beta Flexiviridae, Gamma Flexiviridae and finally Thymoviridae. So, this is all about the 6 orders that were clubbed together in the 2011 ICTV database. Next, we will discuss that how the naming of the families are done in ICTV approach. So, basically for naming of the families, they have selected 4 different criteria. Number 1 is the type of disease that the virus causes. Number 2 is the site of infection of the virus. Number 3 is the physical characteristics of the virus. And finally, the combination of all these 3 features. So, the selection of the family was done on the basis of this all 4 features. Next, we will see that how they are named. The name should be written in capital letters, in italics, and the name should end with a suffix of Viridae. Next we will be discussing about the subfamilies. A subfamily is a group of genera sharing certain common characteristics. A subfamily is usually mentioned in a single name which ends with a suffix of virinae. Next we are coming to the genera, that how genera are written. The genera is written usually in capital letters and italics, but it should end with a suffix of virus. Next we will be discussing about the species. A virus species is defined as a polythetic class of viruses that constitute a replicating lineage and that belong to a particular ecological niche. Usually, the species names are comprising of more than one words. They are usually written in small letters. They are not italicized. Under special conditions, when the species comprises of names of certain geographical locations, then only capital letters are used. In case of plant viruses, we have seen that the naming of the virus is done on the basis of the host and the symptoms that they cause, followed by the name virus. As we have seen the name of a virus as potato necrosis virus, it means this virus infects potato and causes necrosis, and hence the name is potato necrosis virus. Viruses do have names based on the geographical locations wherefrom they have been isolated. As we have virus called Buniam vera virus, which is based on the place wherefrom it has been isolated. Next we will be discussing about the limitations of this ICTV classification. One most limitation of classification of ICTV approaches that they can be classified only when sufficient data in favor of these viruses are already present, means their characterization is being done perfectly and sufficient literatures or journals publications are being done in favor of this classification, then only they can be placed into this taxonomic positions. And when it is uncertain to classify a species into an order, then it is said that it remains an unassigned species in a family. Names are only acceptable if they are linked to a taxa at the hierarchical levels and have been approved by the ICTV. The next benefit of ICTV approach is that it has given space or it has allotted space for the classification of subviral agents. Subviral agents like retrotransposons, satellite viruses, viroids and prions. This was the first time then when we have seen that the subviral agents have been classified according to the taxonomic schemes. So let's have a look on the retrotransposons. Retrotransposons have characteristics very much similar to the viruses. That's why their classification was done just as it was done with viruses. Next comes viroids. Viroids have got certain features common with viruses. They are agents that cause diseases in plants. They basically comprises of RNA genomes, and it has been seen that 70% of the genome remains double-stranded. So these features were known about these viroids, and their naming was done in such a way that we use the suffix viroid for species, we use the suffix viroid for the genera, the suffix virione is being used for subfamilies, and the suffix virioidae was used for the family. So next we will be discussing about satellite viruses. Satellite viruses were again of two types, wherein we have seen that they are called either satellite viruses or satellite nucleic acids. One common feature of satellite viruses and satellite nucleic acids was that they were unable to synthesize the proteins or enzymes which are responsible for carrying on the replication of their genomes. So, they are solely dependent on the helper viruses to provide them with the enzymes which are needed for the replication of their genomes. But there was a difference between satellite virus and satellite nucleic acids, that satellite viruses were able to synthesize certain structural proteins which are responsible for the encapsulation of their genomes. On the other hand, satellite nucleic acids can synthesize only non-structural proteins. They are solely dependent for their packaging purpose on helper viruses. So, this is how we can see that adeno-associated virus can carry on its infection. So, it is a type of satellite virus that depends on helper virus called adenovirus for its establishment and coming out of the host. So, satellite viruses can be classified as subgroup 1 and subgroup 2, wherein subgroup 1 comprises of chronic B paralysis satellite virus and subgroup 2 comprises of tobacco necrosis satellite virus. Next, we look into the satellite nucleic acid viruses. Satellite nucleic acid viruses can be classified as single-stranded satellite DNAs or double-stranded satellite RNAs, single-stranded satellite RNAs, and hence the subgroup 1 comprises of large satellite RNAs, subgroup 2 contains small linear satellite RNAs, subgroup 3 contains circular satellite RNAs. So, this classification is a bit incomplete and it needs to be revised, it needs to be enriched to make it much more effective. But we should be thankful that they have at least taken an effort to classify the subviral agents. The final subviral agent, that is threat of the day, is the prions. So, the prions are famous for causing mad cow diseases. They cause slow, progressive neurological degenerations, and we have seen that they can affect a wide range of hosts, starting from scrapie in sheep. They have caused mad cow diseases in cattle and human infections like kuru. They have also been infective towards Saccharomyces cerevisiae. So, the classification of prions are done basically into two classes. One is called mammalian prions and second are called fungal prions. So, we have seen here that ICTV approach is a much more scientific approach which has taken into account 6 different parameters to classify the viruses and subviral agents both. Viruses have been classified solely on the basis of their own characteristics, their geographical locations, their ability to cause disease, their nucleic acids, their mode of replications. These are the unique features of viruses which were taken into account for the classification of viruses, and really this ICTV approach has helped the virologists to enrich themselves and to get access into the database and to gather informations, and it has really helped them to carry on the study of viruses in a much more methodical and scientific approach.